Electrons are first transported through an electron transport chain, which is a sequence of electron carrier molecules/membrane proteins that shuttle electrons down a series of redox reactions that release energy used to make ATP. Most components of the chains are proteins, which exist in multi-protein complexes 1, 2, 3, and 4. These complexes transports electrons from NADH, which is a reduced form of NAD, to FADH2, which is a reduced form of FAD, until they reduce oxygen. From complex 1 to and on complex 2, Q receives electrons and passes them on to complex 3, which then passes the electrons to cytochrome C, which then passes them on to complex 4. In complex 2, plastoquionin molecules are involved in electron transport in a light-dependent reaction of photosynthesis. The electron releases are high in energy but lose some of their energy as they move through proton complex 1, 2, 3, and 4, which then creates a proton gradient, which is high concentration of protons outside the inner membrane of the mitochondria than inside the membrane, by pumping H+ protons across the membrane into the mitochondrial matrix. At the end of the ETC, electrons are passed to oxygen, which then splits into half, forming water/H2O. The H-positive proteins are unable to travel throughout the inner membranes, so they are forced to move through the special protein called the ATP synthase. It is a complex of several membrane proteins which adjacent electron transport chains using the energy of hydrogen ion concentration gradient to make up ATP. This action causes the enzyme to spin and bring ADP and an inorganic phosphate together to form ATP, which is called chemiosmosis.